Today I will be discussing on the parasympathetic system. In the previous class I looked into the sympathetic system. Here I have placed a picture of Otto Loewi. He won a Nobel Prize in 1936 posthumously. His work was he found out a chemical substance released from the vagus. He named it as a vagus stuff. He did these simple experiments using the frog heart. That means he has taken two frogs and he stimulated the vagus of one frog and collected the output, the heart output from in another beaker or a test tube, and then he perfused it to the other heart, and he found a change in the heart rate. So that was the discovery, and he got the Nobel Prize in 1936. Vagus stuff, now we know that it is nothing but the acetylcholine. Okay, with this brief background about the parasympathetic system, because he gave the idea about the vagus stuff, and the vagus is the major parasympathetic nerve, because it amounts for 80% of the total parasympathetic load in our body. Now, what I did last time, I just mentioned about the various aspects about sympathetic nervous systems, from the transmission to the actions on various organs and to the functions of sympathetic system. I mentioned about the adrenergic agonists and antagonists. Special reference was made to Cannon's fight and flight response. Even I made a brief note on Horner's syndrome. Today, I first briefly provide you the comparison of sympathetic parasympathetic system, events at parasympathetic terminal, the cholinergic receptors, and their the receptor and molecular pharmacology. Very brief, very broad. The parasympathetic actions on various tissue. I will mention about parasympathetic agonists and antagonists. Here what I will do is I will cover the entire autonomic nervous system, drugs influencing the autonomic nervous system. Then I will mention about the functions of the parasympathetic system, and I summarize. So that is my plan of lecture. Now, yes, this is the person, he is with a peace for parasympathetic response and rest and digest. So actually it is the energy conservation. This system works for energy conservation, and it tries to provide or store energy, restore keep energy levels, so that he can go for this fight and flight response. So that means this part of the autonomic wing is consuming or spending energy. This part of the autonomic system conserves energy. This consumes energy, this conserves energy. So that is the difference between these two, sympathetic and parasympathetic responses. And similarly, as we have a fight and flight response, we have opposite of that fight and flight response, that is the playing dead. So especially in some animals, animals like rabbits and other things, if a predator comes, and what they do is they just lie down on the ground as if they are dead. Then once that the tiger or the animal passes away, then they will run. That is the type of response that is playing dead. Now, moving on to the further about parasympathetic system, it is located in cranial nerves and in sacral segments. The cranial nerves 3rd, 7th, 9th, and the 10th. The parasacral segments, second to fourth. This one I have already given a brief idea. I will just read it for you. Synapse at a ganglion located in the vicinity or within the organ. That means preganglionic neurons are longer. They reach the synapse or a ganglion that is very near, close to the organ. The postganglionic fibers reach the effector cells to produce the effect. So again, the parameters of these preganglionic fibers are myelinated B fibers. They are equal to A delta fibers. And the diameter is around 2 to 3 micrometers and conduction velocity is around 5 to 8 meters per second. The postganglionic fibers are myelinated and conduction velocity 0.7 to 2.3 meters per second. The differences in sympathetic and parasympathetic I have shown in this organizational chart. I have already presented to you, and in this organizational chart, so the parasympathetic, you just see that it comes from the brain or the brain stem, cranial nerve nuclei, 3rd, 7th, 9th, and 10th, and the spinal cord. They come here and then to the effector organ. And this one is being influenced by the hypothalamus. Hypothalamus try to govern this wing also. And this hypothalamus receives inputs from the limbic system. The limbic system is in constant touch with the reticular system, cingulate cortex, sensory motor area of the brain. And similarly, the limbic system is monitored by the prefrontal cortex, olfactory cortex, the hippocampus, and many more areas in the brain. All are connected to the limbic system. Just I was trying to make a comment on the playing dead. What happens? You see a tiger there. The rabbit is seeing a tiger, and suddenly the visual inputs that is in the sensory motor cortex, they reach, and these visual inputs go to the sensory association area. From the sensory association area, they reach the limbic system. Now, along with this sensory association area, that would go to the prefrontal cortex. In the prefrontal cortex, the decision is made whether I am going to survive or not. So now the prefrontal cortex says that no, the tiger will eat you or kill you. So then it will say that it will send it to the limbic system. Now it is not the amygdala. Amygdala is an aggressive nucleus that increases the sympathetic activity. It is the septal nucleus. That septal nucleus is activated, and the septal nucleus increases the autonomic parasympathetic activity. That is again coming through the hypothalamus into the parasympathetic thing. The entire muscle tone and the reticular system is activated. The muscle tone and everything will be lowered. Even the respiration becomes slow, and everything as if the animal is dead. That is how it lies down. So this is what the organization as far as the parasympathetic system is concerned. And another important thing I want, before I leave this slide here, that the meditation, what we concern, the meditation increases this activity. So that means you are sitting in one cool place and trying to internalize yourself. And when you are doing that, you are trying to suppress the amygdalar activity. Amygdalar activity is going towards this side, sympathetic activity. Amygdalar activity of the limbic system. If that is amygdalar activity is decreased, the septal activity increases, and the septal activity increases the parasympathetic outflow. That is what we see in yoga and especially the meditation. That is the effect. Now moving on, so this is the organization of the autonomic nerves. You have, this is a central nervous system. These are the neurons in the central nervous system. We have the somatic system, one nerve doctrine, and the parasympathetic system here, the long preganglionic axon and a short postganglionic axon. This is a ganglion which is within the organ or near the target organ. And here the sympathetic system, I have two components there. One, the para or perivertebral chain of nuclei. These are the things, and this is the adrenal gland or adrenal medulla. So that's the adrenal medulla. This red arrow indicates the hormone adrenaline released into the blood. This is the endocrine component. Now, at each level, here, this is all acetylcholine as a transmitter at the preganglionic terminal. Then in the postganglionic terminal, here, in case of a parasympathetic system, it is acetylcholine as compared to this norepinephrine or epinephrine. Okay, so now briefly about, because this is the synapse again, I'm trying to bring in, and I try to call you on different perspective of the synapse. This is one of the synapse, acetylcholinergic synapse. Let us say that synapse at the vagus and the cardiac muscle. Let us imagine that. Now this is the presynaptic postganglionic terminal there, and this is the vesicular uptake or the vesicles. This arrival of the impulse in the presynaptic terminal would make this acetylcholine containing vesicles to reach the target cell and move from interior to the membrane side or the active zone. Once it releases, it produces exocytosis. Released into the acetylcholine is released into the synaptic cleft. This is synaptic cleft. And now this synaptic cleft, depending upon the type of receptors, whether it is a muscarinic receptor or nicotinic receptors, produces the action on this effector cell. Now, this acetylcholine, which is released here, is being inactivated by or destroyed by the acetylcholine esterase. So that means it will be destroyed or it is catabolized into choline and acetate. Now acetate is used in the whatever the TCA cycle as a substrate. So the choline uptake transporters are here in the presynaptic terminal. The choline is taken up, and now this choline, with the help of the energy the mitochondria provides, and an enzyme, choline acetyl transferase. So choline, again in the acetyl coenzyme, you just see that acetate now it is being recycled as acetyl coenzyme, because I said that it enters into the Krebs cycle. So now acetyl coenzyme A, this acetyl coenzyme A plus choline are joined together to form the acetylcholine with the enzyme choline acetyl transferase. Now this acetylcholine is packaged into the vesicles and then put back. Okay, that is about it. Now I have shown one autoreceptor here. This is one of the autoreceptors located on the presynaptic terminal, the M receptor here and the M receptor here. So this is a muscarinic receptor, this is a nicotinic receptors. This is what I have mentioned on the other side. What are the different type of cholinergic receptors? Cholinergic receptors are broadly divided into two, nicotinic and muscarinic receptors. This is a classic pharmacology. Nicotinic receptors and muscarinic receptors. The nicotinic receptors are located on the ganglion. That means a preganglionic, at the ganglion of the autonomic system, whether it is sympathetic or parasympathetic. They are also located in the skeletal muscles. The skeletal muscle, they are the nicotinic type of receptors, and or in the neuronal cells in the nervous system, because in the nervous system again we have a nicotinic and muscarinic receptors located in various parts of the brain areas, basal ganglia, the hippocampus, and even in the hypothalamic areas or reticular system areas. Everywhere they are distributed. And these actually the nicotinic receptors are practically the ligand gated ion channels. or these type of receptors are known as iontophoretic channels. So that means arrival or the acetylcholine sits on the nicotinic receptors, and that opens up the cation channels that produces the depolarization and then the action. That is about the nicotinic receptors. Now we have in the muscarinic, we have here 5 receptors: M1, M2, M3, M4, M5. So this is according to the molecular pharmacology, depending upon the receptor, the molecule, their affinity, their agonists, antagonist interactions, the reactions, and all those things put together, they are classified. Now, broadly, these receptors, because if you are looking at these receptors, when they are activated, they will open up the cation channels. Here, these are these are channels which mediate the reactions with the G protein coupled responses. That means the main ligand is the G protein. Now they sit on the G protein ligand, and then brings about the activation or inhibition. That means a GQ or GI or G1, GO activation. So that means it produces either of the activation that produces this is the signal transduction. That means the M2 receptors, the acetylcholine sitting on these receptors, and muscarine is an agonist and of the acetylcholine. Now acetylcholine is there, it is sitting on these receptors. When they sit on the receptors, that would activate the G protein at GQ, GI, or G1, GO, something like that. That is a molecule that signal transduces. And after that, either there is a activation of the phospholipase C, either there is a suppression of the adenyl cyclase activity, or that is inhibition, or there may be an activation of the IP3 and diacyl glycerol activity that is not shown here. That also would come. So this is about the cholinergic receptors and their actions on the entire effectors. Now, what are the actions of the parasympathetic nervous system on various tissues? I am taking in the same order as I had taken in my last class. Here, first I start with the eye. The acetylcholine produces a contraction of the sphincter pupillae. So that means that produces a meiosis. Meiosis, that is, pupil constricts. If the cholinergic activity increases, the pupil constricts. So that is one thing. So that means when a light falls on the retina, and this light goes to the second cranial nerve, the optic nerve, then optic chiasma, then goes to the pretectal area, then the Edinger-Westphal nucleus comes, and from there it will activate the third cranial nerve nucleus. And this third cranial nerve nucleus is having a motor component and autonomic component. Autonomic component will produce the contraction of the sphincter muscles, that is circular muscles. The contraction of the circular muscles and produce the aperture is decreased, just like your camera. Now that's one part. This is a typical light reflex. Now there is another thing. When we are seeing a distant object and when we are seeing a near object, what happens? There is a ciliary muscle which holds the lens. The lens, you can just see that lens. And when this suspensory ligament, when the ciliary muscles are relaxed, the suspensory relaxed or tense. When the ciliary muscles are contracted, suspensory ligaments slack, and by slackening, the diameter of the lens increases. Especially you can just see that this is anterior and this is anterior diameter, and the diameter of the lens increases. So that means the convexity increases. So that means when I see from distant to near vision, there is a constriction of the pupil That is one component. The second, there is a increased convexity of the lens. That increased convexity is because of the slackening of the suspensory ligament. And this is known as accommodation reflex. Accommodation reflex. So this is a component of the ciliary muscle contraction is by the third cranial nerve. One third cranial nerve contracts the circular muscle, that is the sphincter pupillae. Third cranial nerve contracts the ciliary muscle, and this is necessary for accommodation for the near vision or the relaxation in case of a distant vision. That is one of the part of the parasympathetic action on the eye. Now, second comes the parasympathetic action on the heart. If you the Autolivi experiment, I just again come back. When he stimulated the vagus, it released a chemical, what he named it as a vagus stuff. Now this is known as acetylcholine, and this acetylcholine produces a number of effects on the heart. One, on the pacemaker cell. SA node is the pacemaker of the heart, and we have M2 receptors here, and when vagus is stimulated or when acetylcholine is applied, this activates the M2 receptors. The M2 receptors decrease the heart rate. How that is decreased? That decreases the slope. You just don't worry about that. It is a slope of the prepotential. That slope of the this is the slope of the prepotential. You just see this orange one. This is the slope of the prepotential. So in case of when we apply acetylcholine, this becomes like a flat thing. So if this is a flat, it does not reach this threshold level. This is threshold. This dashed line is a threshold, and it will never reach the threshold. Heart is stopped. So that means so this will monitor the activity of the SA node, and if it is happening slowly, the heart rate is slowed down or decreased. If it is in case of adrenaline, I said that this slope will be faster. This is the rate of rise is faster, that increases the heart rate. And it has this is one on the rate, that is a chronotropic effect. That is a chronotropic effect. Then on the contractility, this is called inotropic effect, inotropic effect or inotropic effect. On the atrial and ventricular muscles, again through the M2 receptors, this is again a GPCR-GI mediated mechanism. They would decrease the contractility, that means through the cyclic GMP mechanism, they decrease the contractility of the smooth muscle or the cardiac muscle, the contractility of the cardiac muscle. That is one. Now, also this is called a inotropism, chronotropism. So then the excitability of the entire cardiac muscle is decreased by the parasympathetic activity. That is excitability is a bathmotropic action. Then we have the conduction of impulses from SA node to AV node to the Purkinje fibers and the bundle of His. So that conduction of through these conducting media, so that is again it's called a dromotropic action, or it increases that this decreases the conduction. So it decreases the conduction. So that means the parasympathetic nerve produces the negative actions on the heart. One, negative chronotropic action, negative inotropic action, negative bathmotropic action, negative dromotropic action. Dromo is conduction, bathmo is excitation, ino is contraction, and chrono is rate. So we have what is called a term, what is called as a vagal tone. It is the tonic activity of the vagus on the heart. A continuous tonic activity of the vagus on the heart. The activity of the vagus on the heart is inhibitory. So that means it is continuously influencing the heart in inhibitory, or it is inhibiting the heart. For that reason, the heart is beating at a particular rate. Say, for example, I have a heartbeat of about 70 per minute. So that means this 70 per minute is because already the vagal activity within me is continuously inhibiting it, and that is why it is 70. Suppose if I block the vagal actions, one interesting thing is you cut the vagus. Okay, when you cut the vagus, the heart rate increases. Okay, that you can do in experimental animals, but in human beings it cannot be done. So under that conditions, we can atropinize. We give atropine. Atropine is a blocker of the acetylcholine at the muscarinic receptors. So when we atropinize, the vagal action on the heart stops or is not present. It is blocked. So once you will get the increase in the heart rate, and that increase is known as a vagal tone. I again tell, now say for example, here is a person with a vagal resting heart rate is 70 beats per minute. After atropine, his heart rate become 150 per minute. So that means a difference 150 minus 70, that is 80 beats, is because of the influence of the vagus, a tonic inhibitory activity of the vagus, that is vagal tone. Now in athletes and sports persons, there is more vagal tone. So that means their heart rate is just slow. You may find a number of sports personalities whose heart rate is just below 60 or 50 or something like that. That is one. I have already mentioned the vagal tone increases with age. And again, as it happens in the elderly people, the vagal tone again decreases, the heart rate start increasing. After 60 or after 65, the heart rate start increasing because the parasympathetic activity diminishes. Personality. Suppose if a person is there all the time restless, worrying, so that means the vagal tone is being modulated by the stress the personality is having, that would increase the heart rate. So that is the personality. Then state of mind, I mentioned about the state of mind. In the state of mind, I mentioned about meditation. If you are cool, you're sitting down, introspecting in a proper environment, so then vagal tone is high. The vagus activity is high. Then species of the animals. Some animals are purely parasympathetic. They are cool animals. They are timid animals. Some animals are aggressive, purely sympathetic. Say, for example, here in our own day-to-day environment, we have these cats and the dogs. Dogs may be cool, so you love their behavior, and the cats they are really aggressive. So the whole entire cat family, they are no different to each other. So that means this is the species of the animal. Say, for example, the tiger, lion, or all those wild animals, they are sympathetic dominance. The rabbits, the deers, and all these animals, especially these are timid animals, and they are parasympathetic animals. So this is the species of the animals. And I mentioned how to determine the vagal tone. This is determined by atropinization. You give the atropine injection, you can determine. Or else you can determine by cutting the vagus. That can be only possible in experimental animals. What are the actions of the parasympathetic system on blood vessels? To make you a very blanket statement, that there are no cholinergic fibers supplying the blood vessels. The blood vessels are all supplied by the adrenergic or sympathetic fibers. The parasympathetic nerve supply is not there to the blood vessels, smooth muscles. You just see that coronary not supplied, skin not supplied, skeletal muscle not supplied, the abdominal viscera not supplied, renal not supplied, systemic veins no. But in case of salivary glands, it produces a dilatation. You must have been taught in the parotid gland or the salivary gland regulation of salivary gland secretion. What happens? This acetylcholine that either the seventh or ninth cranial nerve that releases along with transmitter acetylcholine that releases kinins along with it, or activates kinins. Now these kinins dilate the blood vessels. You know what I'm talking about. It's not acetylcholine per se. It is because of the release of the additional chemical that is a kinins or bradykinin, and that would produce the dilatation. That would produce the sweating, the increased blood supply to the parotid glands, so that secretion continues. So that is about so it's not an exception here. Here it is something in between coming. That is how the blood supply, the parasympathetic supply to the blood vessels. No. Then comes the parasympathetic actions on the bronchial muscles. The bronchial muscles, they produce a spasm or a contraction. Again, I have mentioned here receptor, M2 receptors, it produces a contraction. Then in the stomach, yes, you have the vagus supplying the entire stomach. That would have a number of other mechanisms. One, it increases the motility and the tone of this thing, and that is through the empty, and it produces the contraction. And again, there is another important aspect. There is what is called a receptive relaxation. Receptive relaxation. As with the food enters the stomach, it tries to accommodate more and more food. So that is that is the tonus. The tonus is getting changed, and this is because of the interneuronal activity is altered. Then sphincters. The sphincters are relaxed. That would allow movement. The secretions are increased. So whether it is the secretion of a water, enzyme, or anything, and it will also increase the secretion of the gastrin in case of stomach. Gastrin on one side, histamine on one side, on the other side, and all those products are increased. Same is the case with the intestine. In the intestine, the motility and the tone are increased. The sphincter activity is relaxed. That is the ileocecal sphincter or internal anal sphincters. They get relaxed. The secretions of the intestine, what is called a succus entericus, is increased. So that would increase the water, enzymes, and other things coming from this intestinal glands. Gallbladder, it produces the contraction of the gallbladder smooth muscles. Internal anal sphincter, it relaxes. On the parasympathetic actions on the kidney, ureter, and bladder. So maybe here the uterus also, all the pelvic organs I'm trying to put in. One, urinary bladder, detrusor muscle, it is contracted by the parasympathetic activation. And the sphincters, sphincter vesicae is relaxed. The sphincter vesicae is contracted by the adrenergic components. So that means this opposes the adrenergic. That is what we say as a physiological antagonist. The uterus, its action is highly variable depending upon the hormone profile of the individual, or hormone priming of the uterus, either estrogenic, progesteronic, and that would be on the uterine myometrium. It performs an important functions in the penis, especially the corpora cavernosa. The muscles here are relaxed. They are relaxed. And because of the relaxation of the muscle, there is a pooling of the blood. And this pooling of the blood, because the pooling of the blood makes the erection of the penis. And this erectile response is mediated by the nerve coming from the sacral segment. And this erection is initiated by the release of acetylcholine, and this acetylcholine in turn activating the nitrogen mechanism, nitric oxide mechanism. This nitric oxide produces the relaxation of the corpora cavernosa smooth muscle, and this corpora cavernosa smooth muscle relaxes. The blood accumulates in the interstitium, and that would produce the erection of the penis. So that is one of the important function of the parasympathetic nerve. And this nerve, especially the sacral nerve, is known as a nervi regentis. So this is the nerve which produces the erection. And in contrast, if I were to take the sympathetic component is necessary for ejaculation. Ejaculation, that means the emptying of the seminal vesicle and the emptying of the vas deferens. That is ejection or ejaculation reflex. So that means in the reproductive functions, one, the erectile function or erectile response, and another is a ejaculatory response. Ejaculatory response is sympathetic and erection response is parasympathetic. Both are important here. So if there is no ejection, there may be erection. If there is no ejection, he is not able to produce or reproduce. He is almost impotent. He is nearing to sterility. The erection is an erectile dysfunction. He may be potent. He may have the ejaculation. So these are minor details. We will talk about these things when we talk about the erectile dysfunctions. Now, about the skin, there is no supply to the pilomotor muscles. There is no supply to the sweat glands. Mind you, here the sweat glands receive sympathetic cholinergic inputs. The parasympathetic actions on the liver, no supply. Adipose tissue, no supply, because it don't want to break down the fat. It does not want to enhance the liver activity. And the pancreas, yes, it increases the exocrine activity of the pancreas, so that enzymes and bicarbonates are synthesized, and by that the digestion process is facilitated. So the digestion and assimilation of a food facilitated, the storing of the energy is taking place. Endocrine, it excites the beta cells of the pancreas for insulin secretion, again trying to store the glucose, because insulin increases or allows the intracellular glucose storage. It allows the glucose storing. Then salivary glands, mucus glands are supplied by the sympathetic nerve. The serous glands are supplied by the parasympathetic nerve, and they produce a profuse watery secretion. The serous glands, parotid is mainly serous, sublingual is mainly mucus, submandibular is mucus and serous. The lacrimal glands, that glands which produce tears, they are activated and there is a secretion. That is why you see that whenever you have an emotional outburst, the lacrimal glands they are activated. The emotional outbursts are associated with the sympathetic, parasympathetic modulations, and these sympathetic, parasympathetic activation that would stimulate the lacrimal gland, and lacrimal gland in turn increases secretion. Once there is an increased secretion, our eye chambers are the outside in the conjunctival sac. It cannot hold a corneo-conjunctival sac. It cannot hold the water, and you will have those tears dropping in. That is more secretion. That's a lacrimal gland. So now, how the parasympathetic nervous system mediates its action, and the various receptor subtypes I have already mentioned. Again, I repeat here. So I have here mainly mentioned three type of receptors: M1, M2, M3 receptors. These are muscarinic receptors. These are ligand-gated G-protein coupled receptors. So N1 and L2 are the nicotinic receptors. These are iontophoretic receptors. They open up the cation channels. Now M1 receptors, the pre-junctional endings of the skeletal pre-junctional endings increased acetylcholine release. M2 receptors are located in the lung and the presynaptic parasympathetic nervous system endings on the visceral organs and the blood vessels. All these are mainly the M2 receptors. They inhibit the acetylcholine release. They inhibit acetylcholine release in lungs, whereas in visceral organs they increase the acetylcholine release. Here, the skeletal junctions, they increase the acetylcholine release. In lung smooth muscles, especially the bronchial smooth muscles, M3 receptors produce bronchoconstriction, that is contraction. Then the peripheral parasympathetic nervous system and sympathetic nervous system ganglion, they have these N1 receptors. This would produce a response and a ganglionic blockade. And the N2 receptors are present in the skeletal muscle. That is the neuromuscular junction. Neuromuscular junction is the N2 receptor that would produce the muscle contraction in case of the somatic muscles. Now, the parasympathetic system as a sensory are carrying the visceral sensations. Similarly, the sensations of the buccal mucosa, the entire oropharyngeal components, and the up to the vagus, they are carried by the glossopharyngeal nerve and vagus nerve. Now, here again, these are carried by the vagus nerve. The parasympathetic components, you can just see that it is beyond this line, especially the distal, because it will supply up to this mid of the transverse colon, and subsequently is coming from the S234, S2 parasympathetic S234, and this one, and the trigone region, the prostate, urethra, and all these are supplied by the S234 segments. Now we have the in case of the uterus and the cervix and the upper vagina are supplied by the sacral 2-4. So this is the level of the nerves supplying the different organs, visceral organs by the parasympathetic nerves. That is what I have mentioned. Now coming back here, broadly, this is parasympathetic division. I try to just to summarize. In the ciliary ganglion, it is coming through the third cranial nerves. It produces a dilatation, constriction of the pupil, and it produces the accommodation. That is a contraction of the ciliary muscle to increase the curvature of the lens. And this seventh and ninth cranial nerve, and they will through the otic ganglion or pterygopalatine ganglion, they will supply these salivary glands and the lacrimal glands. They are secretomotor. Then through this 10th cranial nerve supply the lungs, the trachea, and the 10th cranial nerve, the vagus, supplies the heart, all parts of the heart. Then remaining part of the vagus that will supply the stomach, the liver, the pancreas, other viscera of the abdomen. Then they will supply up to the level of the transverse colon here, transverse colon. Then remaining part, the sacral segment will take care of, or the terminal part of the large intestine, rectum, and the anal sphincters. And then we have the bladder, and they are controlled by the sacral segments. Now, the penile cavernosa muscles are supplied by the pelvic nerve. That is necessary for the penile erection. So on this side, we have the sympathetic system. Just you just see the opposite of that. Here it is dilatation, it is constriction. It increases the secretion, it decreases the secretion of tears or the whatever. So then we have it supplied to the from the thoracolumbar outflow. It supplies the lungs, heart, the liver, the intestine, the adrenal medulla. Adrenal medulla is the major. So this is what broadly about the supply. And one interesting thing that the blood vessels do not receive parasympathetic inputs. The functions of the parasympathetic system, I just want to summarize here, or just to give you the broad idea. There may be more functions anyway. So these are one can think about. It is essential for the conservation of energy. That is a rest and peace. And the parasympathetic system is for protecting the animal. So certain animal creatures they are playing dead, or even you will your activity will slow down and you will go to what I will say sleep, or you will be sleepy if your parasympathetic activity is affected. So then there may be somnolence. That means tiredness, or you feel that you don't want to do any activity. That type of a thing that's coming here, the playing dead. It is parasympathetic system is essential for the accommodation reflex for a near and a distant vision. So that means the curvature of the lens through the ciliary muscle contraction and relaxation, and necessary for preventing the excess entry of light or the proper entry of light, the pupillary dilatation or constriction, the pupillary constriction broadly. Then regulate blood pressure and other cardiovascular functions. Parasympathetic system. Vagal tone, it is a cardiac activity monitoring and checking. It will regulate the bronchial smooth muscle contraction and respiratory activity. And it will be a secretomotor to all the exocrine glands. That means it secretes it increases the secretion, and it is also produces the movement or the motor activity. Then erectile response, penile erectile response, stimulate endocrine pancreas to release insulin, produces somnolence. That is coming up here. Somnolence is going to sleep or not being activated. Regulates smooth muscle function, including the sphincteric activity of the entire viscera. Sensory function, that means afferents from the cranio and sacral parts. So from the thoracic and some up to the esophagus level. Up to the esophagus level, they are buccal, pharyngeal and esophageal things are carried by sensory, then pelvic areas by the craniosacral parts. These are the functions of the parasympathetic system. Briefly, I go here, drugs that affect autonomic transmission. Drugs that affect the neurotransmitter synthesis, hemicolinum and metyrosine, they will affect the neurotransmitter synthesis. And here, hemicolinum is a acetylcholine terminals, metyrosine is a norepinephrine terminal. In this case, it blocks the choline uptake, slows the synthesis. In case of metyrosine, it inhibits the tyrosine hydroxylation and thus blocks the synthesis. Then neurotransmitter storage, visamicol and reserpine. These are two things. Visamicol, it will be on the vesicle of the acetylcholine nerve terminal. Vesicles of the acetylcholinergic. This is in the cholinergic nerve. Reserpine is in the norepinephrine or catecholaminergic nerves. And what happens? This visamicol prevents the storage of acetylcholine, whereas reserpine prevents the storage of norepinephrine and catecholamine. Neurotransmitter release, that means that means norepinephrine, dopamine, acetylcholine, prostaglandin, and amphetamine. They all interfere with the neurotransmitter release, especially these norepinephrine, dopamine, and acetylcholine, prostaglandins. They are situated presynaptically. These are presynaptic autoreceptors. They inhibit is located on the cholinergic or norepinergic nerves. They modulate the neurotransmitter release. Neurotransmitter uptake mechanism. The cocaine and tricarbolic acid, TCA, they would increase the uptake. This happens in the adrenergic nerve endings, and they inhibit uptake and prolongs the postsynaptic actions. That means whatever the actions. That is why you will see that the person who is consuming cocaine, he may go into fits and convulsions, and they may die of some excessive adrenergic activity. Many of the sportsmen, they are using cocaine for improving their performance. That is a doping. Neurotransmitter inactivation. These are the drugs. These are edrophonium, neostigmine, physostigmine, DFP, parathion. These are known as organophosphorus compounds. These are organophosphorus compounds. Parathion is the whatever this mosquito repellent and other things are mosquito. The heat and the whatever the drugs you use. These are the things. So what they do is they inhibit the acetylcholine esterase, and this inhibition of these are reversible inhibitors. These are irreversible inhibitors. So this acetylcholine inhibition inhibits the acetylcholine esterase and prolongs the postsynaptic action of the acetylcholine. Now, coming back with the adrenergic system, adrenoceptor activators, alpha-1 receptor phenylephrine, alpha-2 receptor clonidine, beta-1 receptor dopamine, beta-2 receptor albuterol, ritoradine, salmeterol, terbutaline. And their site of action is on the sympathetic postganglionic fibers, blood vessels, piloerector muscles, the radial muscles of the eye, where they will have the pupillary dilatation, the heart muscles, the bronchial muscles, and uterine smooth muscle. Their mechanism of action is through the increasing the IP3 diacylglycerol cascade, that is an alpha-1 receptors, or decreasing CAMP adenyl cyclase activity at alpha-2 receptors. In case of a beta, it is increasing CAMP and adenyl cyclase activity. Adrenoceptor blockers, these are phenoxybenzaimine, generic, non-specific, or specific at alpha-1 prazosin or terazosin, at alpha-2 eohambine, beta-1 and beta-2 propranolol, and beta-2 atenolol and esmolol. So they will act at the